Right. All right, welcome everybody. Thank you all for being here for the Arkansas Native Plant Society's October webinar. This webinar is being recorded, and afterwards the recording will be uploaded to our YouTube channel. Our mission as the Arkansas Native Plant Society is to promote the preservation, conservation, study, and enjoyment of the native plants of Arkansas and to educate the public regarding the value of native plants and their habitats. If you'd like to learn more about us, you can do so by visiting our website at anps.org, or you can check us out on Facebook at facebook.com/Arkansas Native Plant Society. I'd also like to encourage you to join the Arkansas Native Plant Society. You can do that on our website using a PayPal account, just by going to anps.org/join, or you can mail a check along with a membership application to our treasurer, Kate Lincourt, and her address is there on the website on that same page, anps.org/join. The next two webinars coming up in our series are Wednesday, November 17th at 12 p.m. Jennifer Ogle will be presenting on the history of the University of Arkansas's herbarium. And on Wednesday, December 8th at 12 p.m., I'll be finishing up our year's series with a webinar on the edible and medicinal plants of Arkansas. Our speaker today is Dr. Travis Marsico. Dr. Marsico of the Arkansas State University is an evolutionary ecologist whose research focuses on biogeography, biodiversity, conservation, community ecology, natural history, and species invasions. Dr. Marsico is currently researching risk associated with hitchhiking plant propagules at U.S. shipping ports, invasion of herbivorous insect pests, plant diversity patterns and fragmented landscapes of the Mississippi River Alluvial Plain, and plant diversity patterns along elevation gradients in the Neotropics. His research emphasizes making and utilizing natural history collections in research. And today, Dr. Marsico is going to speak to us about the important role of herbaria in recent discoveries of invasive species risk from hitchhiking propagules at international seaports. We ask that all questions be entered into the chat, and we will have Dr. Marsico answer them following his presentation. So without further ado, we'll hand it over to Dr. Marsico.
Thanks, Eric. I appreciate it. I appreciate being invited, and I'm glad to speak with you all today. I did want to ask one question before I get started about the format and the timeline. Is this supposed to be ending at noon or slightly before?
Yeah, typically it's from 11 to 12. If you go a little over, a little under, that's fine. So
okay.
It's not a hard deadline or anything.
And then I also wanted to, before I forget, I wanted to ask too, if you guys are familiar with or connected on the listserv that Diana Soteropoulos from Arkansas Native Plant from Arkansas Natural Heritage Commission sends out. We have upcoming this month a transcription blitz through We Dig Bio, where we work with historical herbarium specimen label data and transcribe those specimen data in a web interface to help Arkansas herbaria become digitized. And I don't know if you guys are already familiar with that and on that listserv and it goes out, but it's something that directly helps us as curators in Arkansas herbaria, and I think it would be in the interest and purview of the Native Plant Mission. If you don't have access to there are going to be webinars and talks, and there's an event, I think, at Natural Heritage, and then there's a 4-day blitz of transcribing specimen data. So I can share that after the webinar.
Yeah, that'd be fine. Or if there's an email that you can put in the chat that people can copy down, that they can reach out to, that'd be another way to get that information out there.
Okay, I have it as like a Word document attachment from an email. So Eric, I might just send it to you, and then you can distribute it as if you think it's appropriate or as needed.
Okay. That sounds great. We can do that.
All right. So I'm really glad to be able to talk to you about this topic. I'm going to be focusing on research that my collaborative team and I have been conducting for the last 5 years. And then as we move forward, we have a currently funded project and we're writing for more grant proposals to extend the research. I'm going to talk a little bit about the future, but I always start my talks on this with a land acknowledgement for where the research was conducted. This is really important to consider that we were conducting our research on land that used to be inhabited by and has important cultural aspects to the Muskogee or Creek Nations and Yamasee Nations in southeastern Georgia. And also where I identified the plants that I'll be talking about, I was at Arkansas State, which is the historical lands of the Osage, Quapaw, and Sioux Nations. The indigenous people of North America were displaced by European colonization. And I want to take a moment to have you guys recognize with me the continued impacts of a colonial mindset and policies on indigenous communities as they continue to struggle for self-determination and sovereignty. If you are interested in this, there is a really cool interface called the Native Land Map that maps the historical native lands of indigenous peoples all over the world, with particular focus in North America, Canada, and the United States and Mexico, and then also in Australia. And then there's also Honor Native Land to learn more about this as well. It's also not lost on me. I just recently had my DNA test from 23andMe, and I am 100% European. Shout out to the Polish and Lithuanians out there. It's not lost on me the irony of giving a talk about species invasion, and as a European descendant in North America and giving the land acknowledgement. I think that's a poignant point to make. But if we look at how hyper-connected our world is today, and we have to acknowledge it is. I mean, one of the things, it's certainly not my research area, but we all are familiar with the COVID-19 pandemic. The speed with which the virus traveled around the world in the outbreak sites, first in China and then moving to South Korea and Italy and these hotspots that broke out and to the US, it's really amazing how fast we move shipping products around the world and how fast we move ourselves around the world. But if you're not familiar with global shipping, we bring in to import sites all over the coastal portions of this country from everywhere else in the world. So just as a hypothetical example, we might be bringing in imports from Europe, Africa, and Asia, and they all are coming to many locations, but those specific ships are going to single locations, like shown here is number 1, the port of Savannah in Savannah, Georgia. And the speed of movement of cargo at shipping ports is tremendous. I've actually talked to some truck drivers recently, and admittedly it's been slowed down because they actually have a lack of drivers. And so that's some of the reason that products get backed up and lost, so that we run out of particular items on grocery store shelves. But under more normal times, an entire cargo ship can be unloaded in a matter of hours, and the individual containers are being moved for containerized cargo is being moved out. For example, the number 1 here is in Savannah, Georgia, and it may get all the way up to Minnesota by a semi truck within 48 hours of arriving at port. Some incredible rates of movement. Occasionally, due to issues with non-compliance or contamination, a ship is turned back. So that's showing what in number 2, my mouse is sensitive here, showing number 2 right here, where a ship comes to port and is actually turned away without unloading due to having some kind of contamination. Hold on, sorry, I wanted to check the chat. Oh, it's all the links for different things. Okay. So we recently, I'll walk through some of the important parts of this figure, but I think if you just like take a step back and look at it, the one element to gain from this is it's complicated. It's complicated to work in an industrialized environment as a botanist because for a few reasons. One, typically botanists don't think of hyper-industrialized areas as places for research. The developed environment is not a place that you think of working in nature. And also, there are lots of actual restrictions to physically getting into the sites. So ports are heavily secured, and there are real and perceived barriers to conducting research in there as well. So we put together a team that partnered with private industry, that's the Georgia Port Authority, and we had the work was funded by the USDA Forest Service. And we had the university researchers, the federal researchers at the Forest Service, the regulatory agencies, which included Customs and Border Protection and USDA APHIS, Animal and Plant Health Inspection Service. And we worked together to conduct floristic inventories and sample shipping containers in order to understand invasion risk at these sites. And the goal was to bring these partners together to improve and increase biodiversity conservation, improve our national quality of life, and build win-win partnerships. So we published this work just last year. Okay, here is a map of our research site. So the Port of Savannah is about 13 miles or 18 or 19 kilometers in from the Atlantic Ocean on the Savannah River. You can see it labeled here as GCT. That's the Garden City Terminal, which is a container terminal at the Port of Savannah, which is just northwest of the city of Savannah, Georgia. It's in Chatham County. It's right on the border between Georgia and South Carolina, as you can see here. That's the Savannah River splits the states. It's a southeastern port. It has a subtropical climate. It does freeze annually, but the idea that it doesn't stay cold for long and might have a high invasion risk is something I'll get into more detail later. That's where we did our research sampling. Here's an aerial view or a satellite image of the Garden City Terminal specifically. And what you might be able to see here is the river is on the right side of the photo. The ship docking terminal is right here. And then you just see stacks and stacks of containers all throughout this property, rows and rows and rows of containers. So 99% of the area is impervious surface, paved land. But there are these little green strips. They're highlighted here in yellow color, but little buffers for water overflow, rain runoff drainage, and there are these green spaces. And my research team and I were nervous that these green spaces could possibly be reservoirs for non-native invasive species if invasive plant seeds were attached to the shipping containers. So we sampled these in a floristic inventory. And what we found over 4 sampling trips in each of the major seasons were that we collected 174 species in 130 genera and 51 families. And of those, 113 species were native, which was 65%, and 61 species, or 35%, were non-native. What's interesting is that we also found 4 state records or species that had not been recorded in Georgia before, and 24 county records or species that had not been recorded in Chatham County before. And they were in a 1-to-1 ratio or 50-50 for native to non-native. So that, I don't mean to overinterpret here, but if it's 35% non-native as a whole, but the new stuff or species that haven't previously been recorded are 50-50, it's indicating at least that the ratio might be increasingly non-native species as time goes by. Which again, I would say that shouldn't come as a shock, being that it's an international shipping port and gets a tremendous amount of activity from all over the world. But with respect to one of our interesting finds, and a story where the herbarium is highly relevant here, is we were making vouchered specimens from our collections that are now available online and shared from this research. And we have to make the permanent specimen mounted and accessioned into our collection with a barcode, and then we image it and share it online and have all the database. And we found what we identified as Glandularia tenera. The Glandularia is a moss vervain, I think, or maybe, yeah, moss vervain as a common name. And it's used, there are many species that are originally from South America that are used in horticulture. But these little projections off the top of the stamens exerted beyond the mouth of the corolla, associated with the anther, that's the part that produces the pollen, and you see all those little tiny white globules there, those are the pollen grains. This is called a connective. Having the connective extend beyond the anther and outside the corolla is one of the few but important diagnostic features to recognize this species. Well, to find, oh, and I've been working with an undergraduate student in my lab on this research. Okay, so what I have here is this is a map based on the Biota of North America, or BONAP, of the distribution of Glandularia pulchella, and I have a question mark by it because I need to discuss some of the complications in the taxonomy, basically throughout the entire southern US. And then, hold up, sorry guys, one. Just made Eric the host again. Okay, so
sorry, I appreciate that. Yeah, I got it figured out though on our end, so I appreciate it. I can help with managing the people now.
Yeah, you bet, no problem. This species is what I'm calling Glandularia aristigera. It's originated in South America, and it has a pretty wide distribution as a non-native species throughout the southern tier of the US. But Glandularia tenera, interestingly, is known from a single county in Alabama, and also, maybe not coincidentally, a port county, right where the port of Mobile is. And so that got me thinking, do we have something really interesting here where Glandularia tenera is not common in the US, but it's coming in at ports, or have some of the plants that have been attributed as Glandularia pulchella or aristigera across the whole southern US, is tenera actually more common? It's just been misapplied. When you look at some of the taxonomic work that has come out of Brazil, you see that there is a different seed shape on the aristigera at this long extended point in tenera, and that connective doesn't extend nearly as far, and certainly not outside the corolla on this other one. And so we got specimen loans from Herbaria from across the country that have specimens, and we've been looking through to determine what we might have here. But an interesting point is, when you're studying non-native species, if you don't have the resources in your region to put a correct name on the species, you're going to be limited by the resources you have available. And so, I don't know, that maybe that wasn't very clear. So said another way, Glandularia is a genus of many species in Brazil and Argentina and southeastern South America. When a new species from that region is introduced here, the botanists who are working in North America don't know what all the options are. So they're going to stick a name on a plant that is the most appropriate, closely known name of something that they know or have reference to. And that runs the risk of having misidentified species in collections because we didn't know what all the options were to begin with for identifying what those plants are. So that's what one part of our research is continuing to pursue, is to resolve some of this taxonomic ambiguity of non-native glandularia here in the US. And answer this question is like, Glandularia tenera really only a port exclusive species? Because that would also be pretty weird. Okay, on to some results from the rest of our floristic survey. When we compare, in this first panel, we compared our research against 27 other floristic inventories that had been conducted in Georgia and South Carolina over the last 30 years. Not all of those studies identified which species were native and non-native. So of the subset of, I think it's 19 studies here, when we look at the percent of non-native species on the per study basis, our study, highlighted here in red, number 28, was by far, we had the highest proportion of non-native species of any of the other locations. Again, this shouldn't come as much of a surprise because most botanists are studying state parks, city parks, natural areas, and areas that you wouldn't necessarily expect to have high levels of disturbance and species, non-native species, and we were working at an active shipping port. When compared based on size, we had a high number of non-native species per hectare. Yes, we had a very small sample area, but we had a high number of non-native species in that area. Only one other study even came close, and most have a very low number of non-native species based on size. And then when we correlate the total number of species at a site with the number of non-native species at the site, basically the more species you have total, the more non-native species you're going to have. That's pretty obvious. But our little data point shows here with not many species total, we had lots of non-native species, pulling it way far from the trend line. Another way to view this information is through the lens of the coefficient of conservatism. Coefficient of conservatism is a ranked scale from 0 to 10 that is attributed to species based on how disturbance adapted they are, or how much they need really precise, specific growing conditions. And basically what it kind of is used for is a floristic quality index, or for conservation, that if you have a site with species, lots of species up in this 7, 8, 9, 10 zone, that means species are really conservative to that type of habitat. Let's say like an upland Crowley's Ridge bog site. If you only find species that grow only in that habitat, then they're really conservative to that type of habitat, which typically means that that habitat is rare, the species are rare, and that habitat is special and worthy of conservation concern and protection. Whereas at the low end, these are species that are adapted to high disturbance areas, and then going, in this case, going below a 2, 1 is non-native and 0 is actually invasive. And you see that at the Port of Savannah, all of the species, pretty much all the species we found are highly adapted to disturbance. Many of them are non-native, and we don't have any species that are on this high level of coefficient of conservatism. So that kind of ends my summary of the floristic inventory. As a review, we had lots of non-native species in really high proportion in a highly disturbed area, and based on the Georgia classifications of coefficients of conservatism, the species we found were also really adapted to disturbance. So for the next part, we sampled these refrigerated shipping containers are called reefers, and the containers are stacked on reefer racks. They're 4 stories high, and we used Ghostbuster style. We used backpack vacuums to vacuum the air intake grill on the refrigerated condenser of the shipping container. This is an interesting area of the shipping container because they have an air intake grill to protect debris from getting into the fan. And what gets captured on that grill are lots of plant seeds. So we vacuum them off in the vacuum tube. is a nylon stocking, so we could capture all the debris. And then we sorted through all that debris, separating dust and insect parts from seeds. And we worked to identify what those seeds were. So we did this for a particular agricultural commodity coming from South America over 2 years. So this graph is showing, in the upper panel of the graph is our first year. It was a 2015-2016 season that we started in August and went through early February. The black are the containers that we sampled, and the gray are the number of containers that came in on a particular day. And some of those we sampled and some of them we didn't. So you see that we usually sampled only a small fraction of the containers that were coming in, sometimes up to half. Sometimes we were not there on site for every shipment day, but you can see that at least we got a good sampling over the course of the season. Then for the 2016-2017 year, the season was actually a little bit longer. It started a bit earlier and ended a little bit later. And we sampled pretty much every other week throughout the main part of the shipping season. And we were sampling some fraction of the containers that were arriving. So what we found was, in season 1, we sampled 331 containers, or 18% of the containers that arrived. And in season 2, we sampled 297 containers, or 15% of the containers that arrived. And we got over 5,500 seeds in both seasons. That means individual seeds collected from those shipping containers, we were able to sum up and identify them, 1, as seeds, and 2, we put them into what we call the morphotypes. So we weren't really identifying them to species at first, but we instead were identifying them into like groupings that seem to be species. We came up with 27 of those in season 1 and 44 of those in season 2. Well, after detailed identification across both seasons, we found that we had actually 30 unique species. So how is that different? Some of them were based on fruit development. So like a little tiny achene that comes off of an aster might not have been as developed. So a younger seed was smaller or lighter in color, and it was separated as a morphotype, but then subsequently I grouped it together saying it was the same thing. And then across the 2 seasons, we had to match those up. So we came up with 30 unique species. And of those, we included 12 of them in some germination trials from season 2. It was important to do germination trials because one of the reasons our project was funded was that there was a debate between Customs and Border Protection and USDA, US Department of Agriculture, whether or not these seeds were even viable after being dried out on air intake grills across an ocean transport. Well, it turns out they are viable. Out of the 12 species we tested, we had 9 species that germinated, or 75% of them germinated. And then the germination rates within species varied tremendously, but I'll get to that in a minute. We had 5 plant families represented, and mostly they were wind dispersed fruits with contained seeds in families that you would recognize. So the aster family with its dandelion style pappus easily gets dispersed in the wind and sucked up on these containers. We did have some Brassicaceae seeds. That one's a little weird. Those seeds are small though. We had Cyperaceae, those are sedges. We had Poaceae, the grasses, again, with a tuft of some type of awn or plumose hairs coming off of the fruit. Those get dispersed easily in the wind. And then we also had Typha, which is the cattail. So we found, of all of those seeds, we found that 4 species of monocots, or 4 at least genera of monocots, are the most abundant. So Saccharum spontaneum is the wild sugarcane or canal grass, and it's abundantly distributed in the Panama Canal. We found that species most. We have found Typha, like I mentioned. Phragmites has native and non-native genotypes. And then Andropogon are the blue stems, which are mostly native to North America anyway. Those were the 4 most abundant seeds that we found. So we did find them over different seasons. So similar to the previous figure I showed, we've got the years separated, the 2016 year and the 2016-2017 year. And you can see how much more compared than the other species we found Saccharum spontaneum, and it was a very pronounced season that right around mid-November and early December, we had this massive spike of the shipping containers containing those seeds. I should note that Saccharum spontaneum is designated as a federal noxious weed. It is considered high risk for invasion through much of the United States. And Typha, the origins are less certain. Typha has been globally distributed, so some species and genotypes are native to North America and some have been introduced and there are hybrids, so it's a bit more complicated, but you can see that we didn't find, I mean, compared to having Saccharum spontaneum on the same graph, it looks like we didn't find any, they just were in low amounts. Phragmites also had a similar late November spike and interestingly, the peak part of that one seems to have shifted between the 2 years, a bit earlier in 2015 than it was in 2016. And then the Andropogon, another interesting thing, was basically flat in the first year, but we found more of them in the second year, showing that there's probably some variation across years. Okay, another way to look at this is where might there be steps in the establishment pathway that these get restricted. And so, for example, if we look at the influx, that's the number of seeds modeled, accounting for all the shipping containers that might be coming in based on the proportions we found. In 2015 and 2016, there were a large number of Typha seeds, relatively large number of Typha seeds coming in, but based on the escape rate from the shipping container that we modeled and how well they germinated and survived, there would be no expectation of Typha becoming established or reproduced on the site. Whereas in 2016, there were enough seeds, and if the same amount escaped, they would basically come down to nothing in germination. But if only a few of them germinated and survived, based on literature search, you guys know cattails, they can produce so many seeds per individual that you only need one individual to survive and establish in order to get a whole population coming back. So we use these alluvial figures to look at what some of those risks are. And frankly, when you take Saccharum spontaneum, there are enough of them coming in, even at low escape rates, they have high germination rates, high survivorship rates, and they reproduce a lot per plant. So it basically seems like you could get a Saccharum spontaneum invasion at any time. Here's another way to look at these data. And these are based on our actual, this is going through germination and survivorship. So for example, we never had more than 50% of the trials that we ran. We never had more than 50% of the wild sugarcane actually survive. But even with really low survivor rates, and we modeled variable escape rates from the container, because we couldn't estimate that, right? We were vacuuming the container ourselves. So we don't know, just because a seed comes in attached to a shipping container, we don't know if it's going to escape from that container or not. So we modeled from 0% escape up to 10% escape and everywhere in between. And what you can see is, based on the germination and survivorship that we know and the influx that we measured, those three things, when we vary escape rate, anything in the yellow, we had a hypothetical 1,000 sites that could become invaded at the port. If that were the case, even at extremely low, non-zero, but extremely low survival and escape rate, it would only take a few generations for all 1,000 of these hypothetical modeled grid cells to become established with Saccharum spontaneum. Less so for Typha, we had less influx in 2015, more in 2016. And then Phragmites and Andropogon also with low survival and low escape, we should be able to find these establishing on site. So this is where we know Saccharum spontaneum is established in the US already. In southeastern Florida, it is already wild and naturalized. It's cultivated around the University of Florida and is part of a global sugarcane improvement program. And in Southwest Florida, it's cultivated, but it might also be naturalizing outside of its areas of cultivation. And when you see where the Garden City terminal is relative to these areas, yes, this is more tropical, and this is a little less, but it's not far away geographically. And Saccharum spontaneum has been modeled to be able to survive in something like 60% of the United States. And based on the number of seeds we see coming in, the risk of establishment once they're here, and that it's already established in the US, it looks like this federal noxious weed is one to definitely be on the lookout for, because we know that it's coming in at the rate of thousands of seeds per year. And that's just from a single commodity. So here's what's upcoming. That kind of summarizes the work that we have completed. And what we have upcoming is we only know the influx on these shipping containers from a single agricultural commodity, from a single source port in South America to a single import site in North America, going through the Panama Canal. What we don't know is how variable the abundance of federal noxious weeds and non-native plants are at these sites in Georgia from multiple sources around the world and from sources not going through the Panama Canal. So we propose to do additional field collections and sample the bills of lading. That's the basically like the flight manifest of a shipping container, where it goes and where it came from and all that stuff. The estimates of total seed abundance, we don't know how that varies. We don't know how the seasonality varies. We don't know based on different types of cargo because we only sampled one type. So we're trying to still use our same site where we have our working relationships developed at Port of Savannah, but we want to expand to other shipping methods, sources, transportation routes, and throughout the year on other types of commodities, and figure out what the overall pressure of this influx of seeds might be in these other seasons. Another thing that we want to do is genomic risk modeling. So the native range of Saccharum spontaneum is Southeast Asia, but some of the sites, because of their elevation, are considered temperate and some are tropical. And so you could figure that the temperate species are adapted to cooler conditions and maybe not adapted to hotter conditions annually. And those, if they are locally adapted, those local adaptations may impact where they can grow. When you look specifically at the Panama Canal region, you see that almost all of those in the Panama Canal region are tropically adapted, which makes a lot of sense based on its location and elevation. But there are a few populations that are still temperate. And because the Garden City terminal in Southeast Georgia is temperate, we might perceive a lower risk of tropical genotypes becoming established throughout the rest of the United States. But an example of what it would look like to do genomic risk modeling based on the propagule types, if we are bringing in mostly tropical species, you can see that the areas of highest risk might be around New Orleans and the Gulf Coast rather than other places on the East Coast, even though there's a genomic match for at least reasonable threat through much of the US. And then lastly, we want to expand our community composition surveys. So we have only sampled the green spaces within this very narrow concrete range within the Garden City terminal. So we want to expand our botanical sampling outside of areas that we have historically done into more of these green spaces that are marginal to the site. Just south, like starting right here, just south and east of the Garden City terminal is the Ocean Terminal. We want to sample in that region too. And you can see scattered across Georgia, the Georgia Ports Authority oversees other shipping terminals. And we're interested in this Appalachian one, which is a rail terminal hub that brings some of these shipments through. Then simultaneously, since we're particularly interested in what's going on in the Panama Canal region through transshipment, we want to know what species are growing at these ports and where we might get some risks coming in from Panama. Our generalized study design and hypothesis is that right where the bulk of the activity is, we might find more non-native species in any of the green spaces and fewer non-native species in these outer areas, even on port property, because it might be a very small spatial scale over which these have initially spread. And it would help in terms of early detection and rapid monitoring for rare plants. I mean, rare, for invasive plants coming in. All right, so with that, my main collaborators on this work are Emily Bellis here at Arkansas State and Rima Lucardi. She's at the USDA Forest Service Southeastern Research Station in Athens, Georgia. We've worked with a big team over the last 5 years on all facets of this project. And we partner with the US Customs Border Protection, USDA APHIS PPQ, the Georgia Ports Authority and Police, and Georgia Forestry Commission to get this work done. And back to the title of my talk, all of the specimen vouchers of the floristic inventory, all of the seed vouchers for records that we found these species of seeds, and the work conducted on the glandularia research with a specimen loan from, I think, as many as 25 institutions for glandularia. We have specimen loans here. We're working on those. The herbarium, where we keep these natural history collections, are actually a really important part of our invasive species research. And having access to and data sharing through natural history collections is really key. Okay, so that felt like a whirlwind and a race, but that did give me more time for questions, and I'm happy